The release of O2 as a consequence of photosynthesis changed the environment in which cells evolved in is commonly thought to have led to the development of oxidative metabolism. Alternatively, oxidative metabolism may have evolved before photosynthesis, with the increase in atmospheric O2 then providing a strong selective advantage for organisms capable of using O2 in energy producing reactions. In either case, O2 is a highly reactive molecule and oxidative metabolism, utilizing this reactivity, has provided a mechanism for generating energy from organic molecules that is much more efficient than anaerobic glycolysis. For example, the complete oxidative breakdown of glucose to CO2 and H2O yields energy equivalent to that of 36 to 38 molecules of ATP, in contrast to the two ATP molecules formed by anaerobic glycolysis. With few exceptions, present-day cells use oxidative reactions as their principal source of energy. Prokaryotes. Prokaryotes include cells of two domains, the archaea and the bacteria, which diverged early in evolution. The archaea includes cells that live in extreme environments that are unusual today, but may have been prevalent in primitive Earth. For example, Thermophilices lived in hot sulfur springs with temperatures as high as 18 Celsius and pH values as low as 2. The bacteria include the common forms of present-day prokaryotes, a large group of organisms that live in a wide range of environments, including soil, water, and other organisms, e.g. human pathogens. Prokaryotic cells are smaller and simpler than most eukaryotic cells. Their genomes are less complex, and they do not contain nuclei or cytoplasmic organelles. Most prokaryotic cells are spherical, rod-shaped, or spiral, with diameters of 1 to 10 mm. Their DNA contents range from about 0.6 million to 5 million base pairs, an amount sufficient to encode about 5,000 different proteins. The largest and most complex prokaryotes are the cyanobacteria, bacteria in which photosynthesis evolved. The structure of a typical bacterial cell is illustrated by Escherichia coli, E. coli, a common inhabitant of the human intestinal tract. The cell is rod-shaped, about 1 mm in diameter and about 2 mm long. Like most other prokaryotes, E. coli is surrounded by a rigid cell wall composed of polysaccharides and peptides. Beneath the cell wall is a plasma membrane, which is a layer of phospholipids and associated proteins. Whereas the cell wall is porous and readily penetrated by a variety of molecules, the plasma membrane provides the functional separation between the inside of the cell and its external environment. The DNA of E. coli is a single circular molecule in the nucleus, which, in contrast to the nucleids of eukaryotes, is not surrounded by a membrane separating it from cytoplasm. The cytoplasm contains approximately 30,000 ribosomes, the sites of protein synthesis, which accounts for its granule appearance. Eukaryotic cells. Like prokaryotic cells, all eukaryotic cells are surrounded by a plasma membrane and contain ribosomes. However, eukaryotic cells are much more complex and contains a nucleus and variety of cytoplasmic organelles. The largest and most prominent organelle of eukaryotic cells is the nucleus, with a diameter of approximately 5 nanometer. The nucleus contains the genetic information of the cell, which in eukaryotes is organized as linear rather than circular DNA molecules. The nucleus is the site of DNA replication and the RNA synthesis. The translation of RNA into proteins takes place on ribosomes on the cytoplasm. In addition to nucleus, eukaryotic cells contains a variety of member-enclosed organelles with their cytoplasm. These organelles provide compartments in which different metabolic activities are localized. Eukaryotic cells are generally much larger than prokaryotic cells, frequently having a cell volume at least 1,000-fold greater. The compartmentalization provided by cytoplasmic organelles is what allows eukaryotic cells to function efficiently. Two of these organelles, mitochondria and chloroplasts, play critical roles in energy metabolism. Mitochondria, which are found in almost all eukaryotic cells, are the sites of oxidative metabolism and are thus responsible for generating most of the ATP derived from the breakdown of organic molecules. Chloroplasts are the sites of photosynthesis and are found only in the cells of plants and green algae. Lysosomes and peroxisomes also provide specialized metabolic compartments for the digestion of macromolecules and for various oxidative reactions, respectively. In addition, most plant cells contain large vacuoles that performs a variety of functions, including the digestion of macromolecules and the storage of both waste products and nutrition. Because the size and complexity of eukaryotic cells, the transport of proteins to their correct destination within the cells is a formidable task. The cytoplasmic organelles, the Endoplasmic Reticulum, and the Golgi apparatus are specifically devoted to sorting and transport of the proteins destined for secretions, incorporation into the plasma membrane, and incorporation into lysosomes and peroxisomes. The endoplasmic reticulum is an extensive network of intercellular membranes extending from the nuclear envelope through the cytoplasm. It functions not only in the processing and transport of proteins, the rough endoplasmic reticulum, which is covered by ribosomes, but also in the synthesis of lipids, the smooth endoplasmic reticulum. From the endoplasmic reticulum, proteins are transferred within small membrane vesicles to the Golgi apparatus, where they are further processed and sorted for transport to their final destinations. In addition to this role in protein transport, the Golgi apparatus serves as a site of lipid synthesis and in plant cells as the sites of synthesis of some of the polysaccharides that compose the cell wall. The internal organization of eukaryotic cells is maintained by the cytoskeleton. A network of protein filaments extend through the cytoplasm. The cytoskeleton provides the structural framework of the cell, determining the cell shape and the general organization of the cytoplasm. In addition, the cytoskeleton is responsible for movement of entire cells and for the intracellular transport and positioning of organelles and the other structures, including the movements of chromosomes and cell divisions.